In today's video, let's talk about various diagnostic tests in coronavirus infection. As of now, we have like three popular tests. The first one is based on PCR, as in polymerase chain reaction. Polymerase chain reaction. The second one are the most popular thing, rapid tests. Rapid tests, also called as antibody tests. Antibody tests. And the most recent third one, nucleic acid amplification test. This is what the technology, the recently released Abbott ID machines. Recently Abbott released a new testing method which releases the result in 5 minutes, which results accurate result in 3 minutes and the negative result in 10 minutes. This is based on nucleic acid amplification technology. In the previous video we have discussed a little bit more about how the PCR works. Today's video will focus about how the rapid tests or the antibody tests work. As the name indicates, rapid tests can be done very soon, very fast. Fast-F testing is very quick. It does not require much technical knowledge. Ease of interpretation. Ease of interpretation. Less expensive. These are the major factors which make the governments to go for rapid testing. However, rapid testing do have their pitfalls, and one of them being the lack of low sensitivity and specificity levels. Low sensitivity and specificity levels. Basically, all rapid tests are also called as antibody tests. Antibody tests. To know what antibodies are, let's speak about this particular thing. Here we have a diagram indicating the immune system of a human being. Here we have a diagram indicating our immune system. Basically, we have these lymph nodes, tonsils, adenoids, thymus, and lymph nodes. These are different organelles which play a major role in our body immunity. How do they function? Say we have a virus here, it infects a human. Whenever a virus infects a human, the natural response of the body is to produce substances which kill the virus. That is a natural trend. Anything whichever is foreign, which is attacking the body, what the natural tendency of the body is to produce substances which kill the particular organism, and these substances are called as antibodies. And why are we calling them antibodies? Because all these infective organisms are made up of protein. Say this virus the basic component is nothing but a protein, and these are called as antigens. And whenever antigens infect a particular person, what happens is like body in response produces substances which inhibit the growth of the organism and we call these are as antibodies. It's very interesting to see how these antibodies act like weapons against harmful invaders of the body. Basically, there are 5 different types of antibodies like IgG, IgM, IgA, IgD, IgE. So these are the several types of antibodies, but for diagnostic purpose the major important antibodies are IgM and IgG. Why are these two significant? They have a diagnostic significance because IgM are the first antibodies to be produced in the body. First antibodies in the sense, whenever there is an infection with any virus, these are the first antibodies which are seen in the circulation. So IgM are the ones which are the first. These are the first antibodies which will be invading the microorganism and try to kill. And IgG are seen later. So they are seen later in the infection. Usually presence of IgM antibodies indicates an acute infection in the sense infection is very, very recent, and IgG is indicative of chronic infection indicating that it is present for a while. Infection is present for a while. So what we are trying to do in this antibody testing is like we are trying to identify the antibodies for coronavirus. So whenever a person comes with the signs and symptoms of coronavirus, what we do is like we basically take the blood sample. We take the blood sample and we check for the presence of these two. Presence of any of these antibodies in the blood indicates the patient is having is coronaviral infection. He is positive for coronavirus infection. Absence of antibodies obviously does not rule out the infection as of now, but presence of the antibodies will definitely diagnose the COVID-19. Let's see the procedure for testing. Basically what we do in this patient is like we take the blood sample. Here is a sample card. Here is a card. This card is pretty much familiar to any pregnancy card. In the pregnancy card usually we have only 2 lines, but here in this particular card we have 3 different lines. The first line, this one representing for control, and the second one representing the IgG component, and the third one representing the IgM component. So what do you basically do? You basically add serum here, blood or serum in this particular receptacle, and you wait for what? 15 minutes. So in this 15 minutes what happens is like this particular serum from here gradually moves from here to here, in this particular direction. As and when it passes through this particular direction, if you end up seeing lines in any of the region, it indicates the presence of that particular component in the sample. Say if a person is if you end up seeing Say if a person is having only two lines, which is a control line and the M line, it indicates the person is having very recent infection. Recent infection is positive for IgM. IgM indicates acute infection. Acute infection. Acute infection. In another scenario where we see if we see only two lines, if we see lines of in the second scenario, if we see the two lines which are at the C and G levels, it indicates the presence of IgG indicating it is a chronic infection. Either the patient is still having the disease or he may be recovered from the disease in the initial phases of recovery. Chronic infection. If the patient is, you end up seeing all the three lines indicate the patient is having an infection, acute infection, and is in the process of progressing from acute to chronic. It indicates the patient is having the infection and this is progressing from acute to chronic infection. Okay, if by any chance, if you don't see this control line here, by any chance, if you don't see the control line in any of the tests, by any chance, if you don't see the control line in any of the tests, it indicates the test is invalid. For a valid test, you need to see the control line. The basic function of the control line is to identify the validity of that particular test. So the presence of the control line will be seen in almost all the cases, irrespective whether it is an acute or chronic. Absence of control line is something where you need to opt for a second card. So we have three scenarios of here. First one being the presence of first scenario is like you have the presence of acute infection where only IgM is present, and then the second scenario where you have both IgG is present or both IgG or IgM are present. This indicates a chronic infection. Let's talk about the principle of the test. So here we have a strip over here and we have this particular area over here. This is the point of application. Point of application is we collect the sample, we collect the blood of the patient, and what we do, we add out over here. So here in this particular region, what usually the manufacturer does is like he takes viral proteins and fixes them over here. I am representing them as green color just for understanding purpose, but usually they are transparent. He fixes the viral proteins in this particular zone, and again another kind of viral proteins in this particular zone. Like we discussed earlier, viral proteins are nothing but antigens. They're nothing but antigens. And again we have another proteins here. These are human proteins only. This is a control line. This is a human control line. So what we do is like we at the point of application we add some serum over here. So what happens after application of the serum? Immediately, as and when the serum is applied, what happens is like there is a transparent paper kind of thing which absorbs, and it moves, the sample moves from this end to this end gradually. Sample moves from this end to this end gradually. As and when it moves it, when it combines with the antigens over here, it forms a complex over here. it binds with the antigen. We call it as, it forms a complex over here, giving that particular red line, that particular green line. And further it moves on further, and if it binds with the antigens of these antigens also, again we'll see another dark line. Again, on movement of further, it again combines with these proteins also and give another dark line. So basically the dark lines over here are nothing but these are in representative of antigen antibody complexes. So already antigens are placed by the manufacturer here, and the person being infected, body produces antibodies. Actually these antigens are mixed with some coloring substance, so whenever this complex is formed, it produces a color. They're designed in such a way, usually they are transparent, and they produce a color only when this particular antigen antibody complex is formed. So as and when this particular complex is formed over antigen I reacts with this particular area, this particular green line indicative of IgM is formed, and likewise IgG is formed. As it moves on forward, automatically it should be reacting with these are components of human antigen. So antigen, those must be reactive with antibodies, and another dark line is formed as control. Here this is also indicative of another antigen antibody complex. However, the antigens does not belong to the virus. However, these belong to the virus. These belong to the COVID virus. So this is what this basic principle is also called as lateral chromatography. So since the sample is transporting in a lateral way, and this is a particular principle in chromatography where any absorbed substance will be moving from point A to point B in chromatography. So this principle is also called as lateral chromatography. So this is the basic underlying principle of this particular rapid test. So what exactly is happening? We have a point of application here, and upon the point of application, we say if this patient is being infected with COVID-19, so what happens is like he is bound to have antibodies to virus. So what antibodies does he produce? That we'll see upon testing. Once the sample goes from this end to this end, at this particular point of reaction, what happens is like the antigen antibody complexes are formed, and it produces a dark line over here. If it produces IgM, we are indicating it as acute infection. If it produces IgG region, it say it is a chronic infection. So this is the way the rapid test basically works. The main disadvantage of the rapid test is the sensitivity. What is sensitivity? Say if 100 persons are tested, how many times is it diagnosing correctly? How many times it diagnose accurately? In sensitivity, we are taking 100. We know 100 known positive patients. We are taking 100 known positive patients, and what we do is we do this rapid test on this particular patient, and as of now, as per the current sensitivity, they are somewhere between 85 to 90%. Somewhere around 85% sensitivity. On an average, they are coming around 85% sensitivity, indicating out of these 100 positive patients, these rapid tests are picking only 85 patients are only accurately 85 patients or diagnosis. Rest of the 15 occasions it is showing the test as negative. This is very dangerous. Like, see, this person is already having coronavirus infection, and we are using this particular test where it is on 15 times out of 100 it is showing as a negative result. That is what we call it sensitivity. Many of the rapid tests we require very good sensitivities, because it should be picking up the diagnosis almost all the time. And another indication what we need to have for rapid tests is specificity. Specificity in the sense, like, say we have 100 patients who are normal, 100 patients, these are normal patients, these don't have any coronavirus infection. If we do rapid tests on these 100 patients, as of now it is showing like something like 80 to 90% of the times it is showing these people are not having infection, negative, or not having infection or negative, which is really good. But on 10 occasions, even though they are not having infection, it is identifying them as positive. This is what specificity indicates. Any rapid test, these two parameters, sensitivity and specificity, are the two important things which we need to look out. Sensitivity and specificity. Tests which are having high sensitivity and high specificity are supposed to be the better test when compared to tests having low sensitivity and low specificity. So apparently at this stage of time what is happening is this particular rapid tests which are out there in the market are having relatively less sensitivity and specificity when compared to PCR and NAAT, nucleic acid amplification tests. When compared to this is having this. However, they have a very good advantage of rapid testing. If you're trying to screen for a community, we can't take the oropharyngeal swabs of all 10,000 people present in this community, and we can't wait for 5 days for the results to come. So in that particular scenarios, what we are trying to do is we do the rapid testing so that we identify maximum number of cases fastly, and if positive, what we are trying to quarantine them, and if negative, we are keeping them under observation for another 14 days.